All right. So I hope everybody is doing well. A couple of general things before I get started. So if you look on the Moodle site, I posted another sort of updated schedule just with I had told you I had an assignment that I wasn't totally sure where it was going to go, and so I finally kind of settled on where that was going to go. The other thing is I don't know why I was feeling particularly inspired about this class last night, but I was. And so I think I have draft PowerPoints for everything until the next exam made. I didn't post them all because there are a couple places where I wasn't quite sure where I was going to end and like exactly where the cutoff would be between the days. The next exam is on Halloween. I will be sending a like doodle poll, and I'll also be sending all the stuff for review sessions and all that later on. But I think I have all of the slides and kind of have everything planned out until then. There was going to be one paper discussion before then, and I had to move that after. So it's basically just going to be different lecture periods until that time. But updated schedule is posted, and I pretty much have all those slides ready. So like I feel decently confident about that happening. Everything kind of happening exactly like that. Remember that the T-cell development problem set is due this week. So send that to me when you get a chance. And today I'm going to be talking about immune cell trafficking. So last time I mentioned a video that I wanted to show you that I couldn't make work on YouTube. So I decided to figure out ways to be super sneaky and tricky with tech. And here is that video. Maybe. And so basically what I want you to see is I want you to see those blood cells moving through the vessels and just sort of how fast that movement looks for those cells, for cells of that size. And so when I talk about this, as I will talk about this quite a bit today, as a crowded freeway with everyone going really fast, this is really what it looks like. And so when we talk about things like if a cell were to just stop in the middle of the vessel as all this was going on, the idea that it would actually be torn apart by the cells that are moving past it, you can sort of see really what that looks like and how that would go. And you will see that in a future video as well. As we discussed last time, we can't just have this sort of happening in a sort of slow, unregulated way. And so we have to have this three-step process happening in order to get our cells to go to the right location. And so I'm going to really, I always call this a three-step process, and I'm thinking specifically about the steps of rolling activation and arrest. There is this other part called diapedesis that is shown here, which I say way less about because there's way less that we know about how it works. And so when I say the three-step process, I do mean sort of rolling activation adhesion, though we will see diapedesis happening as well. And I spent I spent way too much time on all of this, putting all this together last night. But I don't know why. The next slide should be next, and I don't know why I don't have this one here. Like, what was I thinking last night? But I will just point out that this process, we're really focusing on this three-step process right now because we're going to be thinking about a naive T cell leaving the blood and coming into a lymph node or some other secondary lymphoid organ, which is this vision you see in the middle. But the exact same three-step process is also used for like a neutrophil to go to a site of inflammation during an innate response. It's also used later when our T cell is activated for that T cell to go to whatever site is infected. So this is sort of a pretty general set of processes. We're doing it now because it ties into our naive T cell going to the lymph node, but in fact it's the same set of steps either way. And the basic idea is that we've got to have these steps in order to sort of make sure that we have our cell slow down and move over to the side of the vessel instead of just stopping midstream and getting sheared. And so just to show you sort of a general overview of this process, I'm going to show you just part of this video. And I'm going to hope it works, the sound works. Oh, it's very quiet. So here's cells in a blood vessel, and there's a site of inflammation. If you actually went to the YouTube link, you could hear the audio on this, but apparently you can't hear it now. That's okay. So you can see the cells, you can see a site of inflammation. You can see these molecules called selectins that are really important in this process. And so what you'll see is that the cell first kind of pulls over to the side and starts moving more slowly because of these interactions. And so you can see this sort of over to the side and rolling interaction here. Eventually, we're going to see some additional steps where the cell is going to actually leave the vessel and go into the tissue. And so you can see that happening here. And this would be so much easier if we could hear the narration. So this is this exact process actually happening in a mouse in the blood vessel. You can see it sort of going up and down a little bit, and that's because the mouse is breathing. And so you can see the vessel, and you can see how fast all these cells are going, and then you can see some of these cells will actually have pulled over to the side and be going really slow. And so that's sort of the first part of this step. Then they will sort of eventually crawl out into the tissue, and you can see some of those cells that have crawled out into the tissue here. So yeah, you can see sort of some of them that have fully stopped and are starting to crawl, etc. There's the ones that have crawled out. If I was actually the narrator of this video or could hear it, I would tell you that at some point in the video, right here, they actually put a clamp on the vessel so the blood can't run as fast, and so you can see how jam-packed that is with cells going slowly when they have it clamped, and then they'll take the clamp away, and you can see how everything just starts going crazy fast again, and you can really only see those cells that have started in this process of slowing down. And so we're going to be sort of walking through the specific steps and intermolecular interactions that are happening in this process. If you had heard the narration in the video, one of the things that the narrator would have told you is that this process of crawling out of the vessel that you see in the video and that we're going to be talking about today only happens in veins and doesn't happen in arteries. So this is really something that we're only going to see in different types of veins. One of the reasons why we're talking about this now is because we're thinking about naive T cells and B cells in the lymph node leaving the blood vessel. And so they are going to be leaving through a particular type of vein that is going through the blood vessel. When we are in that specific situation, it turns out that if we look at our lymph nodes or our other secondary lymphoid organs, and we look at the veins that are going through those secondary lymphoid organs, we find out that we see specialized veins in the secondary lymphoid organs that are meant to really help this trafficking process go well. So if we're specifically, I've shown you, kind of 3 general situations where trafficking might occur. If we're in that naive T cell or B cell situation going to a lymph node, then the type of vein that we see is specifically called a high endothelial venule, or an HEV. So you can see my lymph nodes in the left, and you can see that the vein is actually shown as an HEV. So if I talk about, the HEV or the vessel is, HEV is basically the specific vessel in the lymph node that is used for these trafficking purposes. I remember as a student just being like, oh, HEV, high endothelial venule. It's kind of vein, so it's venule. High endothelial, great, whatever. I didn't understand why it was called a high endothelial venule for a while, and I remember when I finally learned, I was like, oh. The endothelial cells are basically the cells in the wall of the vein, and the reason why an HEV is called a high endothelial venule is because they're tall. They're like tall and skinny. And so these are particularly tall endothelial cells. That's why it's called a high endothelial venule. And so these are like super specialized for naive B cells and T cells to be able to traffic there. And so sometimes we're going to see the specific situation of a naive B or T cell trafficking at the HEV. We also will see some other particular situations. So we need to think about our different steps of this process. And the first step that we're going to see is this step called rolling. And so in the step of rolling, what we're doing is we are slowing our cell down and we are pulling it over to the side of the vessel out of the stream. What's key here is that the cell is still going to roll along with the flow of blood. The cell is not going to come to a complete stop. Because we're trying to sort of get that cell slow, we're trying to not shear that cell with the speed of all the rest of the blood that's going by. And so in fact, just sort of having it go slowly and not completely stop at first is really key. The other thing that is going to be really key as we think about these steps is that having 3 steps sort of allows a little bit of quality control for this purpose. It allows us to have a situation where we can have the cell kind of making sure it's in just the right location, because it has to have the right protein-protein interactions or protein-carbohydrate interactions at each of these 3 steps to tell it, yeah, this is the right place where you want to leave. Sometimes when people think about this situation, they think about this a little bit like an area code. At some point soon, I'm going to stop being able to use this as well as an example. But if you think about it, if I'm trying to call a number and I start with 973, that means I'm calling someone in this area of New Jersey, right? If I called 919, that would call North Carolina. So I start with 9, but then I go with like a pairing of other things. So the individual combos like each relate to a different location in terms of those first 3 numbers that you're doing in the phone number. And you basically have, a bunch of different choices for interactions here, a bunch of different choices for interactions here, a bunch of different choices for interactions here. And by pairing those 3 things, just like we pair the numbers in an area code, the cell can be like, okay, this is definitely the right spot. I'm in the spot, I'm gonna go. So that's also kind of a key for this, is making sure the cell is kind of slowing down, starting to check if it has the other correct proteins to know that this is the right place, and not getting sheared by the flow of blood happening. And again, in our highly quiet video, wait, maybe it's not going to work, but So here, it's the same video that we were looking at before. You see that the first thing that will happen is that our cell will first start rolling. And that will be mediated by these interactions with a type of protein called a selectin. And so you can see those selectins that are going to have interactions with carbohydrates. And that is pulling the cell over and slowing the cell down for this rolling process. So you can see this here, that the rolling process is mediated by selectins. We have a selectin protein. It has the name selectin because it is a lectin as a type of a protein, which, as we've seen before, is a protein that binds to a carbohydrate. Shockingly, the ligand for this is a carbohydrate. And so there's some particular carbohydrate that binds to our lectin, or our selectin. And so here you can see an example of a selectin binding to a carbohydrate that is specific for. There are 3 different selectins. L-selectin, P-selectin, and E-selectin. And so I'm going to show you some specific examples with L-selectin, P-selectin, and E-selectin, and they do each have their own unique carbohydrate ligands that they will bind. One thing to be aware of when we think about selectin binding to carbohydrates in this situation is that the interaction between selectins and carbohydrates is relatively low affinity. And it's important that it's low affinity. If this was a high affinity interaction, when that cell had this interaction, when we had selectin carbohydrate interaction, the cell would stop completely. And if the cell stopped completely, it would probably get sheared. It needs to slow down first. So we don't want a really strong interaction right here. We want a weak interaction. And so basically what happens is that the cell sort of gets pulled over by this weak interaction, but the interaction isn't strong enough to stop it completely, so it keeps going. And as you saw in the little video, and as you'll see again in a second, there's sort of a patch of selectin. And so the cell kind of gets slowed down and stays rolling slowly in that area. You can kind of imagine the selectin carbohydrate interaction as being a little bit like Velcro. It's like sticks and catches, and sticks and like sticks and comes off, and sticks and comes off, and sticks and comes off, and sticks and comes off. And so the cell is basically sticking and coming off the selectins. It's still getting pushed by the blood, but it's going slower now because it's doing this stick, come off, stick, come off, stick, come off, stick, come off of this low affinity Velcro interaction with the selectin. And so that's really important that we have this sort of weaker interaction right now so that we don't have that cell come to a complete stop. You can see kind of this image here. So you can see we sort of have a patch where we're going to have the right molecules for interaction. And you can see that the cell will get, will sort of get pulled over. This won't be strong enough to sort of hold the cell there. The blood flow will keep pushing the cell, so the cell will keep moving along with the blood flow, but it's just going to keep catching over and over and over again on these selectins, which are basically pulling it over and slowing it down for this rolling step. And this is going to allow the cell to kind of have some further events happen to potentially now stop the cell, now that it's already going slowly, if the cell is in the right place and if we have the rest of the stuff happening. So it's sort of like the cell's moving over, slowing down, checking things out to decide whether it wants to come to a complete stop and whether it wants to actually crawl into the vessel. And so you can also see this in this next video, which again, I'm desperately sad is not going to have the audio that I want with it. So this is a video that's actually really cool. It's called The Inner Life of the Cell. It's an 8-minute long video. I'm just going to show you a couple snippets of it. But honestly, it shows all of cell biology. And it is actually animated using crystal structures. And so this is really what everything looks like. Like if you want to know what microtubules look like, if you want to know what like the mitochondria look like, if you want to know what the gold, like all of it is in this video correctly. I'm only going to show you some snippets, but the link for it is up here. And so what we can see in this video is that we can see, the inner life of the cell. So here we can see a vessel. You can see a bunch of blood cells going really fast, and you can see some white blood cells rolling along the surface. And if we zoom in, that rolling along the surface is happening because of selectins binding to their carbohydrate ligand. And you can see that that interaction is basically like this catch, release, catch, release, catch, release type of thing, where we're getting that cell slowed down and pulled over to the side. Yes, Jazz?
About how different selectins
That's what I'm doing right now. So I was just telling you about this in general, but now we can also be specific to our 3 different situations. Is that what you're asking, Jazz? We can be specific to our 3 different situations and sort of which selectins are involved in which situations, exactly what's going on. So if we first look at this case in the middle where we have our naive T or B cell going into a lymph node, the way that that works is that the naive T cell has on its surface L-selectin. Basically, once a T cell comes out of the thymus, it has L-selectin on its surface because it would like to go to lymph nodes and check for microbes. So just by the virtue of being a fresh new T cell from the thymus, a T cell has L-selectin on its surface. And the HEV, the high endothelial venule, which you can see here, has the ligand, the carbohydrate ligand that binds to L-selectin, which is called CD34. It's also sometimes known, well, it has another name too, but whatever. So what you can, and so HEVs always want naive T cells and B cells to come there. So they always have CD34. They're just covered in this carbohydrate. And as a naive T or B cell comes by, that naive T or B cell will start rolling in the HEV because it kind of would like to go there. Okay? So this is what we always see in the naive T cell HEV situation. We've got L-selectin, which I've got listed in this little table at the bottom, though I've covered up so much of the table that it doesn't really look like a table anymore. You also can see L-selectin on this T cell here. You can see CD34 on the HEV cell here. L-selectin is also known as CD62L, just in case I mess up and call it CD62L by accident, because CD62L and I are old friends. So let's imagine now for a second a situation where our T cell is activated. So this T cell that we're seeing is a naive T cell. Let's imagine now that this T cell is activated. And that activated T cell now wants to go to a tissue. You know, if it's activated by hepatitis B, it might want to go to the liver. If it's activated by SARS-CoV-2, it wants to go to the lung. Whatever it's activated by, it wants to go to that tissue and deal with the pathogen. One of the biggest things that happens when a T cell becomes activated is that the T cell stops making L-selectin. So now, when that T cell becomes activated, it no longer makes L-selectin. Why does it matter that that T cell does not make L-selectin now? Yep, David.
If you're targeting a specific place, you don't want to get caught up in all these other lymph nodes.
Yeah, so now this T cell is no longer going to go to a lymph node. It's going to go somewhere else. And so the idea with the activated T cell is that the activated T cell loses CD62L so that it doesn't go to the lymph node anymore, it goes somewhere else. I'm not going to tell you anything else about the activated T cell situation. So this is my activated T cell slide. This is my activated T cell slide. You can see the naive T cells are in red and they like to go to lymph nodes. Once they get activated in the lymph nodes, they're out and they don't come back to lymph nodes because they have lost CD62L. And the idea here is that they want to go somewhere else other than the lymph node. I'm not going to tell you more specific details than that for the activated T cell. And the reason why I'm not going to tell you more details than that is because there's lots of somewhere elses. And I don't want to go through every single somewhere else in the entire body. So all I want to focus on with the activated T cell is that the activated T cell does not have L-selectin, so it's going to go somewhere other than the lymph node. And so this is one way that our activated and our naive T cells are different, is that they're going to have proteins that are going to allow them to go to different places. If we're in the situation where we have a neutrophil going to the site of inflammation, generally what we see in this situation is that the neutrophil has the carbohydrate, which is called PSGL1, while the inflamed site has P-selectin and E-selectin. And PSGL1 is a carbohydrate that binds to P and E-selectin. Very important philosophical question. Is a neutrophil ever not a neutrophil? Does it ever stop being a neutrophil? Like, not really. Like, a neutrophil is always a neutrophil. It always wants to do neutrophil things. It always wants to go to sites of inflammation. So it has the carbohydrate all the time because it always wants to go to a site of inflammation. Just like the HEV has the carbohydrate all the time because it always wants naive B cells and T cells. So the carbohydrate in both of these situations is all the time, 24/7. It's on a different partner in the two cases, but in both cases that partner never wants to do anything else. However, is a tissue that is inflamed always inflamed? No. So the idea is that when your tissue is normal resting tissue, that tissue doesn't have P and E-selectin. It's not calling in neutrophils. But when you start to have inflammation happening, you can see here's my inflammatory cytokine coming into that layer. Now that cytokine is going to turn on P and E-selectin, And now this tissue is going to start calling neutrophils, which it wasn't doing before. So in both cases, you notice that the selectin is the thing that gets turned on and off when cells want to go to a particular location, while the carbohydrate is on all the time. For some reason, I forgot my white box covering up the rest of this table. Pretend everything other than the rolling part isn't here. So you can see it's basically the same situation going on for all of these things in terms of rolling and how rolling is working. Once our cell has started rolling, it's time for the next step. And basically the idea with the next step is going to be another type of protein-protein interaction, which is going to be important for a couple of reasons. One of the reasons why this protein-protein interaction is important is because it's another way of making our area code specific. It's sort of the second number in our area code to help us know, oh yeah, this is the location I want. If, for example, the slowed down neutrophil or the slowed down T cell doesn't get this interaction, it's going to just roll for a while. It might run out of selectins and it might go back into the bloodstream. But if it gets this second protein-protein interaction, it's like another checkbox that says, yes, this is the right place that you are going to want to leave. So that's one of the reasons why this step is really important. But there is another reason why this step is really important. And this step is known as activation. And so now we're going to see this activation step happening after our cell has started rolling. If our cell can't roll, and there are actually some people who have mutations where their cells don't roll, although I think it's been found in like 2 people in the history of the world. There are, weirdly enough, a lot of cows that have the mutation. Then their cells basically never start this whole process. They can't do activation because they never did rolling in the first place. Activation happens via signals from a chemokine. And so the interaction that we're seeing in this case is interaction between a chemokine and the chemokine receptor. And remember that chemokines are proteins that are secreted by different cell types. They're basically, a specialized type of cytokine because they're a cytokine that involves cells moving, right? And so we'll have this secreted protein shown as like the blue circle in the top that will bind to some kind of chemokine receptor. And you can see a little bit of the details of the chemokine receptor and chemokine receptor signaling here. Not only is this really important in terms of just providing the second area code spot for the cell, this cell also is going to get some important signaling information from this chemokine. And so the other reason why this activation step is important is because the cell actually has to get activated and get some information from the chemokine signaling. So you can see our chemokine binding to a chemokine receptor here. Notice the chemokine receptor structure is shown. The chemokine receptor has a specific kind of structure. It's actually different than any cytokine receptors. The chemokine receptor has 7 transmembrane domains. I mention this because later in the semester, I'm going to talk about a particular chemokine receptor and a mutation in it. And so I want you to know that a normal chemokine should have 7 transmembrane domains and look like that. And chemokine receptors are a type of receptor that you learned about in Bio 250, which is called a G protein coupled receptor. So when you learned about G protein coupled receptors, They had 7 transmembrane domains. They had a structure that looked like this. And they also had some of these partners called G proteins. That's why they are G protein coupled receptor. So chemokine receptors are G protein coupled receptors. You can dive into signaling of G protein coupled receptors. But what I want you to note is that when we get some signaling through that chemokine receptor, a lot of events happen in the cell. And so you can see that we're going to get some changes to the cytoskeleton. We're going to get some changes to differentiation, proliferation. We're going to get a lot of signals into this cell as a result of signaling through the chemokine receptor. Immunologists have a specific kind of naming convention for chemokines and chemokine receptors. It is based on the structure of the protein of the chemokine. And so chemokines all have some cysteine residues in them. And those cysteine residues can have different numbers of gaps in between them. And so some of them will have a structure that looks like this, and we'll call that chemokine, we'll say CC. Some of them will have a structure that looks like this and say CXC. Some of them will have a structure that looks like this, XC. Some of them like this, CX3C. Basically, you don't need to know those specific structures or things, but if you see a name, CXC something, CC something, XC something, CX3C something, that should ring a bell in your head, chemokine. And particularly, if you look, we will often see names that have things like CXCL, CCL, XCL, CX3CL. If it's an L, the L stands for ligand. So if something has one of these names with an L in it, it's a chemokine. It's the little blue circle guy. If you see the exact same kind of naming, but instead of an L, you see an R, then we're talking about a receptor. The most famous one that I'm going to tell you about later in the semester is called CCR5. So CCR tells me it's a chemokine receptor, and it's chemokine receptor for this type of structure, number 5. And similarly, I'm going to tell you at one point about one called CXCR4. It involves this structure, it's the receptor, and it's number 4. And so if it has one of these types of names and an R, then we're talking about the receptor here. And so all of our different leukocytes have a lot of different types of chemokine receptors. So differences in chemokine receptors are big in indicating part of this address code for the cell and where exactly that cell should go. So this is part of the way that the cell knows, oh, I'm this type of cell and I should go to this location. And so you can see that, neutrophils versus basophils versus eosinophils versus monocytes will have some different chemokine receptors. You can see a resting T cell versus an activated T cell will have different chemokine receptors. Those will send each of these cell types to different locations. So this, in reality, when I think about that activated T cell going to lots of different potential places, I often think about this as being really dependent on what kind of chemokine is involved. I think of the chemokine as sort of really telling the T cell which place it's going to end up in. Because there's only 3 choices for selectins, where there's like 50 choices for chemokines. Our chemokine is the next step of this process and to have this chemokine activation. And so in the case of our naive T cell, the naive T cell will have on its surface a chemokine receptor called CCR7. And CCR7 will bind to the chemokines CCL21 and CCL19. So again, this is how our naive T cell is like, okay, I'm a naive T cell, I have CCR7 on my surface. The HEV is like, I always want naive T cells, I'm always going to make CCL19, 21. So we're always going to have this kind of interaction happening. Remember our activated T cell? And remember how I told you that one of the key things about the activated T cell is it loses L-selectin, so it stops going to a lymph node. The activated T cell also loses CCR7. So again, we're basically telling that cell, you're grown up now, don't go to lymph nodes, go somewhere else. Go to a different tissue. Again, the details of which chemokines are going to be important here will vary based on which tissue, but we lose CCR7 expression because we no longer want that cell to go to the lymph node. And so you can see that the CCR7 is the thing that's getting turned on and off based on where the T cell is in its life. The CCL19 and 21 are getting made all the time by the HEV, because it always wants to bring in new naive T cells. We can also think about exactly what's happening in the case of the neutrophil going to an inflamed site. The only problem with the neutrophil going to an inflamed site is that one of the names is annoying. So in the case of the neutrophil going to an inflamed site, the inflamed site starts making the chemokine. So the inflamed site starts making the chemokine, which is called IL-8 because it got named before we made up this whole CC naming. Like, we found IL-8 really early, and so it got IL-8 as its name, even though it's not a cytokine, it's a chemokine. It's just, I think that it also is CXCL8. Like I think that is also its name, but I always call it IL-8 here. And the neutrophil will have the IL-8 receptor binding to IL-8. Neutrophils are always going to have the IL-8 receptor because neutrophils always want to go to an inflamed site, while normal tissues do not make IL-8. But when that tissue starts to be inflamed, that tissue starts to make IL-8 to start to call in the neutrophils. And in fact, if you go back to looking at some of the information about innate immune responses, you will see IL-8 show up as one of the cytokines that was being made. So now we get to our next step that's going to happen during leukocyte trafficking. I have heard this step be given multiple different names. So this image calls the first step rolling, and it calls the second step activation. If you were listing the steps for me and you called the second step like chemokine signaling or chemokine activation, that would be fine. The third step I've heard called different things in different places. So sometimes I hear the third step called arrest. Sometimes I hear the third step called adhesion. Sometimes I hear the third step called firm arrest. The other thing you could call the third step is stopping. This is where you come to the complete stop. This first step of rolling, sometimes I've heard called as sticking. So sticking versus stopping. And so now we're going to have that cell that's been going along come to its total complete stop. So I want to think about this idea of the cell coming to this total complete stop, whether we're calling that arrest or adhesion or stopping or whatever you want to call it. I want you to think about it for a second. I've already told you a little bit about the interaction in molecules that we see with rolling. And I told you something super important about the interactions that you see in rolling. What did I tell you about those interactions? Yeah, Jazz? Low affinity. It was super important that they were low affinity. Why was it super important that they were low affinity? Yeah, Paige. So we didn't stop the cell completely. Why would it be bad if we stopped the cell completely? Yeah.
It could be sheared by the other blood cells that are flowing.
Yeah, it's basically going to get torn by the other cells. If our cell stops too soon, if our cell stops before it's pulled over and before it has slowed down, it's going to get destroyed, right? And so it was really important that we had that low affinity interaction first. Now at this step, we're going to see stopping. If you had to guess about the interaction that we're going to see with stopping, what would you guess is going to be a key feature about that interaction? Yep, Lucas. It's going to be high affinity. Now we are going to have something that's going to be high affinity to really completely stop that cell so that it doesn't go any further in the blood vessel, so that it can actually crawl out of the vessel. So we're going to have some interacting partner that we haven't talked about yet. And whatever that interaction is going to be, it's going to be a high affinity kind of interaction. But I want you to think one more step about whatever this partner is with its high affinity interaction. What happens if that high affinity interactor does its interaction too early? What happens if just by accident one time that interaction happens to a cell that's going about its daily cell life? And suddenly this interaction protein just happens to bind its ligand randomly because it just did. Because sometimes there are just random protein-protein interactions that happen. What would happen if this interaction happened randomly to a blood cell? Yeah, Abigail. It would get sheared. Suddenly our cell that's not supposed to be stopping would pull to a halt when it wasn't supposed to and get sheared, right? And so what you should notice or should realize about this interaction that I'm going to tell you about for the third step is that this third step interaction has to be pretty tightly regulated because it can't happen early. And in fact, what we see here is that we're going to see interactions with a protein that can come in two different forms. Most of the time, when the cell is just chilling, the protein is in a form that doesn't really bind to stuff. However, if the cell has been given a signal that says, yeah, things are looking good, this might be a stopping place, then the cell is basically going to uncover this protein so that it can stop. And so at the beginning, this protein is not ready to stop the cell because we don't want to stop randomly. We only make sure that this protein is ready to stop after we kind of have already checked, yeah, rolling's good, yeah, activation's good. Okay, now we're going to check for the third step and we're going to actually turn on the protein to do that checking because we didn't want that protein to be available too early. That change in the protein that we see for step 3 is the other key part of step 2. So the other thing that is happening with our chemokine signaling, when we have this chemokine receptor and we're getting this signal in the cell, this also is going to tell the cell, make the stop protein ready now. Get the stop protein ready for stopping. And so step 2 is also really important because step 2 is what preps the protein we need for step 3. And that conformational change is happening because of a signal that comes from the chemokine receptor. So is this protein that undergoes this change, is that protein going to be on the naive T cell or the HEV? This protein that's going to get changed as a result of signaling through the chemokine receptor. Which one's it going to be on? Which one? The T cell. You want to know why it's going to be on the T cell? Because that's the one that had a chemokine receptor that could get a signal. So the signal comes through this receptor. So whatever the protein is that we're going to see make an important change must be on this cell, because that's where the receptor signaling can happen. What about in the neutrophil inflamed site situation? Where's this changing protein going to be? It's on the neutrophil, because that's the one with the receptor. And so notice that it's not always the selectin and the receptor and things on the same cell. It differs in different situations. But one thing we can always say is this protein that changes conformation is on the same cell as the chemokine receptor. And so now this protein that I feel like I've been hyping up so much is a protein that we see here, and it's called an integrin. And so we're going to now see the action of an integrin. Integrins have 2 different protein chains. They are generally referred to as an alpha chain and a beta chain. Different integrins will have different alpha and beta chains. What we now know is we now know that there's like a few choices for alpha and a few choices for beta. And so if I were to discover a new integrin today, I might name it something like alpha 4 beta 8 integrin. Just which alpha and which beta it has. Some of them that were discovered a long time ago, like this one, have other names because it was before we came up with that naming convention. So like you can see LFA1 is here. But oftentimes, as you will see on this next slide, the integrins are alpha something, beta something. And yeah, they do. Many of them have other names because we figured them out a long time ago. But you can see all of these different integrins. And again, just like with the chemokine receptor situation, the particular integrins are usually associated with particular anatomic locations. So when I read a paper about certain integrins, I know, I'm like, oh, that T cell is going to go to the gut because that's the gut integrin and things like that. There's sort of a bit of a link here as part of our area code. The integrin ligand, you can see here. You could also see on the previous slide, is known as it has some name that has CAM in it. You can see CAM here. CAM stands for cell adhesion molecule. There's a bunch of different things that are something CAM. And so I'm just going to say we've got integrins, they bind to CAMs. You can see some of these examples here. So we've got ICAM, VCAM, MADCAM, we've got a bunch of things, but they all CAM, so that's all you need to worry about. So we've got integrins binding to different types of CAMs. And that's going to allow for our stopping to happen. So the other key thing that you need to know about integrins is what goes on with them in terms of conformation and conformational change. So at the beginning, integrins are not ready to bind. They're not ready for stopping because we do not want to stop our cell too early. And so I'm going to show you the crystal structure and sort of about what integrins are like. I'm going to tell you the name of this conformation, and I'm going to do a silly interpretive dance. But integrins start out in a position that looks like this. It is actually known as the jackknife conformation. Think of a Swiss Army knife, where the knife part is bent down in the thing. So integrins start out like this. And they can't bind to things because their hands are the bindy parts. So they start out like this. You can see that's them starting out like that. When the cell gets a signal through a chemokine receptor, the integrin goes into an extended conformation. You can see an example of it here from one textbook, an example of it here from another textbook as well. And so basically, your textbook shows it in like a weird way. It looks like floppy and then like makes a new shape. That's not good. That's not real. This is really what it looks like. Really, the integrin starts out like this. When the cell gets signal from the chemokine receptor, it goes like this. And is now ready to bind to CAM. While it was like this, it was unable to bind to CAM. And so integrins have to get activated in order to bind to that CAM. And you can see this integrin activation process here. You can also see this integrin activation process here. There's a chemokine binding to a chemokine receptor. Now we've got some there's my G protein coupled receptor. Some signaling happens. Here's my integrins that are bent over into the jackknife conformation. And now there's going to be a signal, and ta-da! The integrins are going to extend out and now be able to bind to the CAM molecule that they were not previously able to bind to. And so now our cell is first going to roll, then it's going to stop, and finally undergo that process of crawling through in between the cell layers. And so you can see that process happening here. But none of that is possible until we see the cell stopping. And so in the case of our naive T cell going to the HEV, you can see that the HEV has a CAM called ICAM, while the naive T cell has an integrin called LFA1. So again, it's the exact, we've got the integrin on the same cell as the receptor, binding to the CAM on the HEV. And in the case of the neutrophil, oftentimes, in fact, it is the same LFA1 ICAM interaction, though it can be some other types of interactions. And so again, you can see there is one shared partner here, just like in 919 for North Carolina and 973 for New Jersey. Both of them use a 9. So we have one place where we sort of have a shared partner and one place where we have unique partners giving us those address labels for these different locations. You can also see that the integrin is on the same cell as the chemokine receptor to allow for that integrin to be activated. Once our cell has stopped, our cell is actually now able to crawl through the blood vessel wall. And so you can see that process of crawling through the blood vessel wall happening here. You also saw that in the earlier movie. This step also I've heard called multiple different names. One of those names is diapedesis, which you can see on this slide. The other two names are written out on the next slide. I don't often talk about this one as like its own special step in as much detail. And the real reason why nobody talks about this one in quite as much detail is because we don't really understand how it works as well. It's really hard to actually like model in a dish cells crawling between other cells layers in a vessel. And so there's a lot that we don't really understand about exactly how this process works. Though we do think it is very much driven by a lot of chemokines. So we think chemokines are very important. As you could kind of tell from that other video, and as you will see in the next slide, this cell has to undergo some pretty massive shape changes. So you saw it like spread out and get skinny, and then actually like push through another layer of cells. So there's a lot going on in terms of kind of the cell biology, the cytoskeleton, things like that here, but again, we don't know a ton about the details of that. And so here you can see, here is a leukocyte, here is a cell wall layer, and here is that leukocyte basically crawling around that cell layer. So you can see that cell like dramatically changes. This process is sometimes called transendothelial migration, because we're going across, so transendothelial, across the endothelium, and we're migrating. Sometimes it's called diapedesis. Sometimes it's called extravasation, extra outside, vasation, vascular, going outside the vessel. So all three of these names, as far as I'm concerned, are totally interchangeable. And you can see this process happening here. You can also see this process happening in this comic from this immunology comic person who I really like. So here you can see, the blood vessel wall, and you can see basically the lymphocyte really does just sort of squeeze through this tight layer of cells and get itself to the other side. It's pretty important that that layer stays really tight and it's just sort of squeezing through. You don't want your blood vessel wall to become leaky and leak everything out. You just want that one individual cell to sort of crawl through, kind of like you see here. And so those same steps are the steps that we're going to see in all three of our situations of leukocyte trafficking, of our white blood cells going to different locations. One thing to also point out, one thing that I have learned much more previously is that there are probably a lot more things regulating this process than we originally thought. One thing that I've learned about more recently is it turns out cells actually are able to traffic better, leukocytes are able to traffic better when you have a fever. It seems like a fever actually changes some of the trafficking proteins on cells. So it might not be that the fever is just about, raising your body temperature to kill microbes. It also might be doing things like allowing better trafficking to different sites and things like that. And so there have been more and more situations about things where I would have told you no, that isn't involved in immune cell trafficking, where we suddenly see that this trafficking process gets messed up in a bunch of weird situations. And so there's probably a lot more perhaps going on than we necessarily thought. This is one paper telling us that, in fact, fever promotes T cell trafficking. And so when you have a fever, your T cells are better able to traffic. So the reason why we are talking about all of this right now is because we are following the life of our T cell. Remember that T cell has finished doing T cell development in the thymus, has left the thymus and is in the bloodstream, and potentially wants to find its antigen and actually make a response. We're basically going through all those steps of the T cell making a response. So we are really looking at this T cell going into our lymph node in order to initiate an adaptive immune response. That's why we talked about this whole process now. When that T cell comes into the lymph node, as you see here, it doesn't just go any old place in the lymph node. The T cell actually goes to a specific location inside of the lymph node. And that location is the T cell zone. And so here you can see I've got a spleen and a lymph node. They both have T cell zones. So here you can see the T cell zone of the spleen. Here you can see the T cell zone of the lymph node. And so we have T cell zones. And the T cell will come into the lymph node and will specifically move to the T cell zone. The reason why the T cell is able to go to the T cell zone is that there are more chemokines in different places of the lymph node to direct the cells into different locations. So basically, once the cell gets to the lymph node, it is going to continue responding to chemokines that are going to move that cell to the right location. In the case of our T cell, we're going to the T cell zone. Yep.
So they go to the T cell zone, almost like registration, and then at registration they tell the T cell where to go thereafter?
No, so it comes in, like it's in the HEV, like registration, and then it comes in the HEV and it's like, you are supposed to go to the T cell zone. Okay, does that make sense? Yeah. So there are sort of chemokines around that are like signs that are being like, this is where you go. And so they're pushing that cell towards the T cell zone. We've already seen the T cell zone, or I've already mentioned the T cell zone to you a little bit last time. So there's also something else in the T cell zone at this point. In fact, that something else got to the T cell zone by following chemokines that told it to go there. Same thing. Something else came into the lymph node and got pushed to the T cell zone because of the chemokines. Do you remember what else we saw going to the T cell zone? Yeah, Abigail. So our dendritic cell that we saw last time that came to the lymph node that has picked up antigen is also hanging out in the T cell zone. So our dendritic cell with antigen got pushed to go to the T cell zone, and the T cells are getting pushed to go to the T cell zone. And so now we've done the first part of the process of why the heck it takes so long for an adaptive immune response to happen. We got the cell in the right location, and we got the antigen in the right location. Transport of antigen. Getting the cell into the secondary lymphoid tissue. Now we're ready for the T cell to recognize the antigen, because they're both in the right place. Now we can actually have that first interaction between that T cell, which is a naive T cell who's never seen antigen before, at least never seen foreign antigen before, and that dendritic cell. And this is happening in the T cell zone of the lymph node. And that's going to sort of start out this process that is eventually going to get us to this good adaptive response. And so basically what we're going to see is we're going to see our dendritic cell that's going to interact with our T cell. And you can see that interaction happening here. What you can notice is, again, there's sort of a bunch of steps. These are actually all these steps that are happening in this image are the things that are happening during this time. So now we're basically zooming into this block of time. We're zooming into, okay, so we got the dendritic cell and the T cell in the T cell zone. We're going to start to see an interaction, and we're going to see some additional events. And all of this is basically what we're doing between now and the exam, is we're looking at the specific events that are going to happen. With the dendritic cell, remember that a dendritic cell is a professional antigen presenting cell, so it has MHC class II on its surface, potentially. It also is a nucleated cell, so it also has MHC class 1 on its surface. It can have both. So it could turn on a CD8 or a CD4. This image helpfully shows us both just because it's easier to have all of those things happening. And so we're going to see some initial interactions that are going to happen with that cell. That interaction is going to largely take place via the T cell receptor on the T cell interacting with MHC plus peptide on the antigen presenting cell. Remember, the T cell receptor needs its partner CD4 or CD8 and also needs its partner CD3. Remember, CD3 is bringing the ITAMs, CD4 or CD8 is bringing the kinase. And so we're going to be able now to see a lot of different signal transduction events happening in the T cell that are going to lead to all of these changes in the T cell that you see here. And so on Friday, I'm actually going to zoom in here, and we're going to go through the signal transduction events. Friday is like hardcore signaling day in terms of looking at the signal transduction events that are happening in the T cell, which things are phosphorylated, which things are cleaved, all that kind of stuff, and what changes that makes to the T cell to eventually get all of these other events to occur. And so I will see you guys for that on Friday.